good evening. I hope everyone is doing well. Welcome to our latest installment of the Torrey Botanical Society's Spring 2023 lecture series, our Torrey Talks. My name is Jordan Hoffman. I'm a professor of biology at Eugene Lang College at the New School, and I'm also a member of the Torrey Botanical Society Council. And so from the founding of Torrey Botanical Society back in 1867, one of our main activities has been hosting these lectures on a variety of topics in botanical research. We've hosted some really nice talks over the last year, and you can check them all out at our YouTube page, actually. It's youtube.com/Torrey Botanical Society. And so we have, a fairly nice backlog of talks for the last couple of years, especially since we've been having virtual talks, since the whole COVID pandemic and everything. So the best way for you guys to stay updated on our future lectures, as well as field trips and other activities here, is to become a member of the society, if you're not already, where you can stay up to date on all these different things. You can find information about how to become a member and what that entails on our website, is torreybotanical.org. And you can also follow us on social media. We have Facebook and Twitter accounts. So our handle on Twitter is @TorreyBotanical. And I'm going to provide just some links in the general chat here. Lydia, thank you for doing that. I really appreciate it. So that's awesome. So that if you want to stay up to date, in addition to becoming a member, you can check us out on any of those social medias. Additionally, once a year, we try to have, we haven't been able to do it because of COVID, but things are improving now. We try to have a yearly in-person talk in March to accompany our annual society banquet. And so if people are in the area, this is open to attendance. It'll take place a weekday in March at the New York Botanical Garden Library, and we're going to provide more details for that when it comes. So that will be an in-person talk, and then obviously an in-person banquet as well. So anyway, so tonight's talk is going to be presented by Alden Dirks, a PhD candidate at the University of Michigan. Alden grew up on the doorstep of the mushroom capital of the world, otherwise known as Kennett Square, Pennsylvania. But at the time, the only thing notable about mushrooms to Alden was the smell of the compost heap. And however, in college, ventures into mushroom foraging led to a fascination with fungal biology for Alden. He began this journey in academic mycology at the lab of Professor Don Pfister at Harvard University, where he participated in fungal biodiversity survey of Boston Harbor Islands, a national rec area. And after that, Alden went on to the University of Wisconsin-Madison to study arbuscular mycorrhizal fungi in association with switchgrass. Incidentally, our next presentation for February, that's the next month, is actually on a similar subject. We're going to be hearing from another biologist about arbuscular mycorrhizal fungi. So sort of following in Alden's research history here. But at that time, he ended up founding the Madison Mycological Society, an amateur mushroom club. And so today, Alden is currently completing his PhD at the University of Michigan, where he's doing research focused on genetics, evolution, and the distribution of Gyromitrin toxin in false morels, which is what he's going to talk about us tonight. Before I turn things over to Alden, I just want to go over a quick few notes about Zoom. So, we can keep our videos on or off during the talk, but I would like to ask that you please keep yourself muted so as not to disturb the speaker. You can enter comments and questions in the chat as you go, however. We do a Q&A portion after the talk is complete. And during that time, I will be reading off the questions from the chat to Alden for him to answer. If you would like to submit your question that way, that's fine. Or if you'd like to ask your question verbally, you can use the raise your hand feature to indicate in the chat that you would like to speak, and we'll give you a chance to do so. If you have to leave early or would like to share the talk with someone else, it'll definitely be recorded and available on our YouTube page shortly, so that will be an opportunity for you to do that. And so with that, I'd like to turn it over to Alden. Alden, thank you very much.
Thank you, Jordan. I will share my screen at this time. Would it be okay, actually, if we did a quick answering a few questions kind of before I get into the results, just from the chat, if anyone has any clarification questions they need to understand the content better?
I don't see why not. I think we can do that.
Yeah, if people have questions that are relevant for the first few parts, I'll just check them real quick at some point.
Yeah.
Just to make sure everyone's up to speed.
Yeah, if you'd like to just prompt the like when it's time to just prompt the group, and then allot a few minutes for people to submit responses, because it can take a little bit.
Okay, sounds good. Thank you. Great. Well, thank you everyone for being here tonight and for having me. My name is Alden Dirks. I use he or they pronouns. And the talk tonight I'll be giving you is about the first chapter of my PhD research. This research was recently published in the journal Mycologia, and it's entitled Not All Bad. Gyromitrin has a limited distribution in the false morels as determined by a new analytical method. Now, before we begin, I would like to share with you an important campaign that's happening here in Philadelphia. I am a graduate student at University of Michigan, but I actually live in Philadelphia and work remotely. And this campaign is in honor of, or I'm sharing this with you in honor of the Lunar New Year. Philadelphia Chinatown is a long existing neighborhood in Philadelphia. It's a vibrant and celebrated part of the city. Residents and tourists come to enjoy food here, as well as recreate and enjoy the culture. And more importantly, it's home to generations of Asian Americans. Unfortunately, Philadelphia Chinatown is currently threatened by a proposed new arena by the Philadelphia 76ers. The 76ers have an arena in the south part of the city, along with all the other sports stadiums, but they would like to build a new one that's literally right next door to Chinatown. And this is expected to have really devastating impacts on Chinatown if it goes through, primarily through gentrification, through rising prices that would displace residents. Not to mention the cost, the traffic and pollution and noise that would come along with 5 to 10 years of demolition and construction. Unfortunately, this is not the first time Chinatown has fought back against proposed projects like this. You can see in their logo on the top right, the word arena is superimposed on stadium and casino. So that is the original logo from the past 25 years as Chinatown has fought back against proposed development and constructions of these baseball stadiums and casino. Unfortunately, before that, a few projects have happened, resulting in eminent domain and displacement and destruction of hundreds of houses in Chinatown. So this work is in support of affordable housing for all and to maintain culturally vibrant cities above the profit-seeking motives of the arena builders. So if you have the means to do so, I encourage you to go to the Asian Americans United website, aaunited.org, and make a small donation if you can. It would really help. So thank you. I'm going to jump into the talk now. I'm going to start out with an introduction on these what I find to be fascinating, interesting, also very weird looking mushrooms called false morels or lorchels. And then we'll get into the methods of my research, which is a sample collection and phylogenetics. And then after that, talk about this new method we developed for analyzing mycotoxin, gyromitrin in these mushrooms, ultra high performance liquid chromatography. I'll then go on to the results, how we validated this new assay, the distribution of gyromitrin that we found in the family Discinaceae, and some new data that we collected since this publication in December. Finally, in the discussion, I'll summarize what we learned from this research. I'll give my opinions on the edibility of lorchels and share some ongoing research that we have related to the genetics and evolution of the gyromitrin mycotoxin in lorchel mushrooms. So let's start with an introduction. And I'll say once more, if you have some questions about the introduction to methods, please feel free to share those in the chat. And about halfway through the talk, I'll look at those just to make sure everyone understands what I'm talking about. Okay, so these false morel mushrooms are iconic primarily for what they're not, which is morels. And here's an image of a morel on the left, what I call a true morel. This is the species Morchella americana. It's a yellow morel. These are in the family Morchellaceae. And these morel mushrooms in field guides are regarded as choice edibles. In the spring, a ton of people go out looking for these, especially in the Midwest. They can only be foraged, they cannot be cultivated, so they fetch a hefty price as well if you can find them in markets. So these are really celebrated mushrooms in field guides, and a lot of people like to forage them. When you're learning how to forage morels, one of the first things you're taught is how to differentiate a morel, true morel, from a quote unquote false morel, also called a lorchel. A lorchel is, the name is not used as commonly, but I'm on this rebranding campaign calling these things lorchel because I want them to be what they are, not what they're not, which is not a true morel. Anyway, so these lorchels, regardless, in field guides are typically regarded as deadly poisonous, and you're warned to stay away from them, to avoid them. They're toxic, they're bad. And these are in the family Discinaceae. This species is Gyromitra esculenta, which is found in Europe. If these mushrooms are so bad, why do people eat them? This is most prominent in Scandinavia, particularly Finland. This is a picture of a marketplace where you see a whole bunch of these mushrooms in some containers, and these are consumed widely in Scandinavia as a delicacy in the spring. However, these are known to be toxic, and they have to be prepared in a particular way to render them safe to eat. For that reason, these have, they've made for themselves the nickname fungal fugu, after the Japanese pufferfish, that if you don't prepare properly, it can kill you. Similarly, with these mushrooms, if you don't prepare them properly, they can make you very sick, possibly kill you if you're exposed to enough of the toxin. On the right is another one of these lorchels. This is the beefsteak lorchel, Gyromitra caroliniana. There is a Facebook group called False Morels Demystified. It's kind of a watering hole where a bunch of people online gather to talk about eating lorchels. And this is one of people's favorite mushrooms that a lot of people eat without any ill effect. So what's going on? How come some of these are okay and some of them are not? Well, the toxin that is found in some of these mushrooms is truly, it's truly toxic. It's not something to be messed with. The toxin is called gyromitrin. It's named after the genus Gyromitra. And if someone is unfortunate enough to be exposed to this mycotoxin, symptoms, they happen in about 5 to 12 hours after exposure. Some common and mild symptoms, I say mild almost in the COVID way, you know, mild COVID was you didn't go to the hospital, but you could still be very sick and you wish you weren't that sick. So mild gyromitrin toxicity includes gastrointestinal distress, vomiting, abdominal pain, and diarrhea. If you're exposed to enough of this toxin, it has neurological symptoms, including confusion and shaking that can progress to full-on seizure in the worst case scenarios. This is because the toxin and its derivatives bind to vitamin B6 or pyridoxine, which inhibits the production of neurochemicals like GABA and serotonin. It can destroy the liver, causing hepatic coma, and in the worst case scenario, these things all progress to death. There's also some potential long-term effects to gyromitrin exposure. research in the 1900s, in the 70s and 80s showed that the toxin seems to have genotoxic effects in animal models that can cause cancer. And there's some emerging research, particularly from 2021, a study that found that exposure to gyromitrin can cause neurodegenerative diseases. There was a hotspot of patients with ALS in the French Alps, and it was found that what they had in common is that they were eating these mushrooms with gyromitrin. So for all these reasons, gyromitrin is not good and it should not be consumed. Chemically, gyromitrin is a small mycotoxin. It refers to principally this chemical called acetaldehyde, N-methyl-N-formylhydrazone. This is the principal chemical, and it was first identified in 1968. Following research in the 70s, identified 8 more higher aldehyde homologs, which are numbered 2 to 9. There are probably other homologs to gyromitrin, even larger molecular weight substances, but primarily when we refer to gyromitrin, we're referring to this first chemical, which is abundant in it has the greatest abundance in a mushroom. Gyromitrin is also a reactive mycotoxin. So gyromitrin and its homologs, 1 to 9 from the previous slide, even at room temperature, especially upon heating and in acidic environments, gyromitrin will break down into N-methyl-N-formylhydrazine, which further derivatizes into monomethylhydrazine. I have this little rocket emoji here because monomethylhydrazine is used in some rocket propellants. So when you eat gyromitrin mushrooms that contain gyromitrin, you're eating rocket fuel. And this rocket fuel, monomethylhydrazine, has been used in industry for a very long time, and we know that it's very bad for you. It's acutely toxic and carcinogenic, and you just don't want to be eating it. However, this reactivity, volatility, and solubility of gyromitrin explains how you can actually eat these mushrooms if you prepare them properly. And the proper preparation includes boiling the mushrooms, rinsing them, and then boiling them once again in fresh water, and then rinsing them, and then they're ready to cook. It's a laborious procedure that people do, apparently because they taste so amazing. I haven't had the opportunity to eat Gyromitra esculenta, but people do a lot of work to be able to eat them. Finally, gyromitrin is a rare mycotoxin. It belongs to this group of mycotoxins or secondary metabolites defined by this nitrogen-nitrogen bond. We call these NN bond-containing natural products, or N2MPs. There's only about 200 of these N2MPs that are known in nature, and this is a very small percentage of the total known secondary metabolites. We have no idea what enzymes make this bond in eukaryotes. We know a little more in prokaryotes. And in general, we just have a limited understanding of secondary metabolic pathways and genomic arrangements outside of a few model fungi. So within this world of rare secondary metabolites, there's another interesting N2MP, which is agarotene. Agarotene, just a fun fact, is a mycotoxin. I don't know how fun it is, but it's a mycotoxin found in common button mushrooms, Agaricus bisporus, that you would buy from the grocery store, like these white button mushrooms in the top corner. And it's also found in other Agaricus species. Fortunately, its concentration in Agaricus bisporus is not thought to pose any risks to human health, but it does share the same NN moiety, and we also don't know what genes make agarotene in Agaricus bisporus. So despite all this, people eating these mushrooms, we actually have very limited evidence concerning which Morchellas are toxic. Within the Discinaceae family, there's about 75 species. It's a paraphyletic collection of stipitate, discoid, and hypogeous forms. Stipitate refers to ones that have a stalk, like the Gyromitra picture on the top left. Discoid species are cup-shaped or saucer-shaped, and these used to be in the genus Discinaceae, but it was found that many of these are closely related to more commonly known Gyromitra species, and a lot of these have been moved to the genus Gyromitra, but Discinaceae is the origin of the family name Discinaceae. And then there's these hypogeous or truffle forms, Hypnotria. And these form groups are all mixed together within the family. And this is a pretty common thing for fungal families. You find lots of different form groups all present together. And that's because there's a lot of convergent evolution in it. And over a few million years, things evolve to look quite similar, and they can evolve to be stipitate or to be a cup again, back and forth or whatever. Within the family, there's only 2 species that have actually been confirmed to produce the Gyromitrin mycotoxin. These are Gyromitra esculenta, which is where the mycotoxin was first isolated and received a lot of research in the 1960s and 70s in Europe. And then there's Gyromitra gigas. However, this one is a little less certain. This comes from a paper from 1980. It isn't a very high quality paper, although the method seems sound. They looked at 3 specimens of Gyromitra gigas, 2 of which had very small amounts of gyromitrin in them. One of them was basically at the detection threshold. And either way, this is like 80 to a few hundred times less than Gyromitra esculenta. So this is the only evidence we have, solid evidence for species that produce gyromitrin. A number of other species are suspected to contain gyromitrin based on poisoning reports. This is the Gyromitra esculenta group. Other species that are closely related to Gyromitra esculenta, as well as Gyromitra ambigua, for which there's 1 documented poisoning from 1976. And then there's a collection of species that are anecdotally regarded as safe. For example, on the Facebook group that I mentioned, False Morels Demystified. And this is by people who eat lorchels. They say that they eat these mushrooms all the time, no problem, they're totally safe and they're delicious. These are the North American Gyromitra gigas group specimen species, and I'll talk about those in a minute, as well as the Gyromitra brunea group. However, an important distinction here is that people who don't eat lorchels typically regard everything as poisonous. And this is coming from more of a toxicology or public safety null hypothesis, which is that you shouldn't be messing around with mushrooms if you're not sure if they're safe. So there are 2 different camps here, and this has led to a lot of contention with some people enjoying lorchels, no problem, and other people saying you shouldn't eat them. So we'll get into that at the end when I share with you my opinions on eating these mushrooms. So I just want to showcase some of the really interesting mushrooms that we're talking about here. Within the esculenta group, we have Gyromitra splendida. This is a species that I have 1 specimen that I've sequenced from the East Coast. All the others are from the West Coast. This is a primarily West Coast species. And then we have Gyromitra venenata. This is our primary species on the East Coast. These have a brain-like cap, and they have light to darker burgundy, almost purple browns sometimes. They're really interesting, strange mushrooms. And then we have the infila group. This is a species I mentioned, Gyromitra ambigua, which we hypothesized to contain gyromitrin. These have these kind of inflated caps. Infila has what's described as a bishop cap kind of cap to it. And ambigua is looks quite similar, but has purplish hues to it usually. And then we have the Gyromitra gigas group of the Northeast. These include Gyromitra americana gigas. This was a species myself and collaborators described as a new species last year, as well as Gyromitra corfii, which is our primary Northeast gigas group species. And then Montana, which is primarily a West Coast species, although one of them has been sequenced from the East Coast. So for a lot of these, don't have a good sense yet of their biogeographic distributions. And then we have the Gyromitra brunea group. These ones are really cool and delicious. I've eaten Gyromitra brunea and it's fantastic. This species has these disc-like or cup-like caps. They don't quite fuse together, giving it the common name elephant ear lorchel, because those discs look like elephant ears. And then we have Gyromitra caroliniana. This is called a beefsteak lorchel. This has more of a southerly distribution. It's a really massive mushroom, and people really like to eat this one as well. So getting into the questions and hypotheses of the study, the overarching question for this portion of my research was, which Discinaceae species produce gyromitrin? People have been asking me this question for 6 decades, and I set out to do my best to answer that question, not comprehensively, but just do our best to get a sampling of the family. For that reason, we set out to do a broad sampling to look at presence and absence, rather than trying to quantify how much toxin was in different species. Our hypothesis was based on published phylogenetic trees and toxicity evidence. I hypothesized that Gyromitrin evolved in the last common ancestor of the Esculenta group, as well as the Ambigua group, with loss of function in this truffle clade, the Hypnotria genus. And that's because as a truffle, your main way of spreading spores is mycophagy. You attract animals to dig you up and eat your fruiting body, and they spread your spores for you. So if you're being spread around by mycophagy, it wouldn't make much sense to intoxicate and kill the animals that are spreading your spores. And this research fits into my larger overarching research questions on the genetics and evolution of Gyromitrin, which include what are the genes that are responsible for Gyromitrin biosynthesis, and how exactly did Gyromitrin production evolve in the family? Great, so let's jump into the methods, and then I'll go to see if there's any questions, just so we're on the same page before I go into the results and discussion. So to do a survey like this, you need specimens, and those specimens came from myself. I collected a number of those. A very important source was donations from community scientists. We're in this amazing age where in the United States, where people are increasingly excited about mushrooms, and there's just an exponentially greater number of amateur mycologists who are willing to share their collections. One person can't do it all. I received specimens from all over the place and very interesting materials. So this work would not have been possible without all the donations from dedicated mycologists out in the field. I also received a number of specimens that were preserved in museums in Fungaria, as well as cultures from culture collections. So our specimens consisted of mushrooms, or the ascomata, as well as mycelium, cultures that we generated from single spore isolates. These specimens, we sequenced the ITS and LSU ribosomal DNA barcodes, and we inferred a phylogenetic tree with these data using a concatenated data set. I have many collections that have come out from this work. They are amazing and they're supporting ongoing taxonomic research. I just want to highlight one of these species that we described as new last year, Gyromitra americana gigas. Andy Miller is leading the charge on revising the taxonomy of this group, and we're using molecular phylogenetics and morphology, looking at one clade at a time, revising taxonomy, and certainly we'll be describing a number of other new species. In terms of the ones that we used that we tested, there's 66 of those, and we did 105 tests. Some of these specimens were tested multiple times, which is why there are more tests than specimens tested. That's because we were curious about how the preparation or the state of the specimen affected the results. In the beginning, we were curious whether, for example, how did Gyromitrin compare between frozen mushrooms, freshly frozen versus dried? If you dry the mushroom, did it lose all of its Gyromitrin? So many specimens were tested more than once. In terms of the actual testing, there have been a number of tests that have been developed over the past few decades. However, nothing in recent times and nothing that's really up to modern standards. So for this research, I partnered with natural product chemists at University of Michigan, and they developed this great new test using ultra high performance liquid chromatography. Gyromitrin by itself is difficult to work with for 3 reasons. One, it's water soluble, so it's difficult to separate from all the other water soluble things. It's a low molecular weight, and for chromatography purposes, it's not good because it lacks a UV chromophore. It does not absorb electromagnetic radiation in the UV spectrum. So to fix these issues, the chemists came up with this derivatization reaction employing 2,4-dinitrobenzaldehyde. When you react these Gyromitrin derivatives with 2,4-DMB, you get these Schiff bases, and these Schiff bases solve the issues with detecting Gyromitrin. They're nonpolar, so they can be separated from water-soluble things. They're larger, and they absorb UV light, so they can be detected with UV chromatography. Here's just a brief diagram of a very simplified diagram of how the test is conducted. You take your starting material, which can be a culture or dried mushroom or whatever. As a negative control, you place some tissue into acetonitrile and water. As the test specimen, you put some in acetonitrile and water, and you add your derivatization reagent, the DMB, as well as trifluoroacetic acid to cause that reaction to happen. You take these solutions, you run them through ultra high performance liquid chromatography, diode array detector, mass spectrometry. And here is just a diagram of how UHPLC works in a very simplified way. So you take your sample and you run it through a column. The column has a stationary phase. This is the chemical that is lining the column. In our situation, we're using a non-polar stationary phase. This means that non-polar chemicals are slowed down as they travel through the column. The tubing separates chemicals basically by polarity. And then your retention time is how long it takes for something to travel through the column. So all the non-polar things will travel through very quickly, and the more polar and larger a substance is, the longer it takes to move through the column, and this allows us to separate out the chemicals. A UV light at the output detects the absorbance of the chemicals that are coming through, and we can map this to a standard to determine the unique signal of the Gyromitrin derivatives. Great, so with that, I'm just going to quickly see if there are any questions.
If anybody has any questions, feel free to write them in the chat here, and Alden will address them in the next couple of minutes here. But yeah, there's one right now. If you, I can read it off for you.
Sure.
So how does boiling remove the toxin? Is it drawn out into the water, or is the chemical reaction? Is it the chemical reaction with the water and heat?
Right. So this is, I think both are happening. It is water soluble, so it is going out into the water. It's volatile by itself. So actually a lot of it's being released with the steam, and some of it's reacting to form these breakdown products. All three things are happening at once. But this is an important question. I don't think anyone should be eating these mushrooms, but if you want to venture into it, I wouldn't stop you. I guess I would if I was by you, but I couldn't stop you. Anyway, but it's important when you're boiling these mushrooms to not be exposed to the steam. This is maybe the only mushroom that you can get poisoned by just by breathing in the steam of the cooking, air, as it gets like burning poison ivy, right? So you don't want to breathe in the steam from cooking these mushrooms because it contains the toxin. Even people who will like travel with these mushrooms in their car, they'll put the windows down as they off gas, so they're not exposed to too much of the toxin.
Crazy.
Yeah.
I have a follow-up question to this. You said also that drying the mushroom eliminates the toxin as well. How long did like are we talking about like a matter of days, or because that obviously must be a serious issue for you when you're doing these collections and you're trying to measure if these things are actually, if the chemical is present in these different collections.
Right. Yeah, I'll get into that. It takes time to, it's kind of an exponential decay reaction. So the longer you let it go, the more it will be lost, but it kind of reaches an asymptote. For that reason, it's not ever entirely lost. It gets down to levels that are not dangerous. And this could be like after months of drying it, basically. Lots of it's lost in the first like day or so. Mm-hmm. But yeah, there were some studies in the 70s where they looked at that, like how it's lost over time. So drying is a big part of how it's prepared to rid the toxic mushrooms of a lot of the gyromitrin. But that wouldn't be sufficient. You would definitely need to boil it and off gas any amount that was remaining.
Okay, thank you.
Yeah, you're welcome.
I don't see any other questions, but if anything pops up, you're welcome to address them.
Sounds great. Yeah, I'll get back to the talk now. Cool. So I'm going to get into our results, starting with the validation of this new assay. Here again is the diagram for what we do. we found that using a pure positive control chemical of gyromitrin, adding this DMB and acid, we saw these distinctive peaks corresponding to these two shift-based derivative products. What this is a chromatograph, and it's showing the absorbance on the y-axis. This is the height of the peaks versus the retention time. So as I was saying before, polar substances move very quickly through the column, which is these big peaks in the beginning, and the non-polar substances take longer to move through the column. So around 4 minutes is how long it takes for the gyromitrin shift-based derivatives to move through the column. This gyromitrin pure standard doesn't really have anything else going on because it's just gyromitrin for the most part. That's why it has these two pure peaks. Comparing this to mushroom extracts from Gyromitra venenata, which we expected to contain gyromitrin, we found the same distinct peaks at the same retention time. And when the acid was excluded from that, those peaks went down quite a bit, which is what we'd expect because acid is important for this reaction to progress. When we took the extract without any of the derivatization reagent or acid, we didn't get any peaks as a negative control. So this is great. We validated their assay, and we did other things to be able to measure quantity and whatnot. So as another validation, we are just seeing how long it takes for the reaction to happen. Running the reaction from 0 hours to 24 hours, more 12 is converted to 13, resulting in these two peaks turning into one peak. And you can see the negative control is absent of any peaks. The first thing we did is we took a bunch of mushrooms from the museum and we said, okay, how old can a mushroom be? Can we take dried mushrooms that are old and still detect gyromitrin? And to our delight and surprise, we actually detected gyromitrin in very old mushrooms. Here on the right, you can see chromatograms going from top to bottom is more recent specimens to older specimens. And we had no trouble detecting gyromitrin in specimens going back to 1945. We even had one specimen from 1883 where it had trace amounts of gyromitrin. So this was really great because it meant that when we dried a mushroom, that was a year or two old, we didn't expect it to lose all the gyromitrin. And at first I was curious about this because it is a volatile substance. And I asked the chemists, how come we don't lose it all? And they gave a great analogy, which is that you can dry herbs like basil, and you can still smell the volatiles coming off that basil. And that's because drying doesn't get rid of all the volatiles. A lot of these volatiles stick around in the dry tissue. Some of them will bind to larger molecules before they're released when you grind them up or whatever. And in general, even a basil that's 5 years old, or even something more pungent like, I don't know, rosemary or something, it can lose its pungency, and over enough time, you probably wouldn't be able to smell those volatiles anymore. However, if you bring in a modern chemistry instrument, it can detect extremely small amounts of these volatiles. So this helped me understand that just because you dry it, certainly it's losing the volatiles, but you're not losing all of them. And that's why our study was focused on presence and absence rather than absolute quantity, because just in the process of collecting it from the field and bringing it back to the lab, we would have no way of controlling for how much gyromitrin was lost in that process. Great, so I'm going to jump into our phylogenetic tree. And just as a brief primer on what a phylogenetic tree is and how to understand this, we have all of our different specimens listed here with their species names, and they're connected together with these branches. And branches come together at nodes. When the more recently or the closer the nodes are together, that means that they share a more recent common ancestor. So when we go from right to left, we're moving back in time basically to the ancestral populations of these specimens that are in our current time. This tree is made with rDNA, ITS, and LSU barcodes. We marked the specimens that we tested for gyromitrin with color. The blue ones are ones that we did not detect gyromitrin in, and the red ones are the ones that we did detect gyromitrin in. For reference, we included other species, and the ones that are bolded are ones that are type specimens. So these anchor our species concepts. Okay, so I'm going to jump into the individual clades, starting with the ambigua group. If you'll remember, our hypothesis was that ambigua contained gyromitrin based on some poisoning reports, and these Hypnotria truffles here on the bottom were absent of gyromitrin. We did not detect any gyromitrin in any of these clades, whether the Infilo group or Spherospora and Californica or the Hypnotria truffles. When we moved to the Esculenta group, we detected gyromitrin in everything. We detected gyromitrin in lots of Gyromitra venenata. A lot of these came from Michigan that I was collecting myself. We also detected it from Antarctica, from the southern hemisphere, as well as some from West Coast species and specimens. And then, to our surprise, we did not detect gyromitrin in any other species outside of that group, except for Gyromitra leucosantha. Gyromitra leucosantha was not on our radar because it's not a species that people eat, to our knowledge. It is this yellow cup-shaped species, and it tested positive in both situations. There was no gyromitrin detected in the Gigas group, nor in the Brunea group, to the excitement of people who like eating these mushrooms. Interestingly, within Gyromitra leucosantha, this yellow cup fungus, not only did we detect these peaks corresponding to our shift-based derivatives, we also found this very strong peak corresponding to an unknown shift-based derivative. This was absent from the negative control sample, and it does not correspond to either of our derivatives that we know about. So this is likely some undescribed secondary metabolite that's related to gyromitrin. And since this publication, I just want to share a little bit of new data that we've gotten. There is another species that's undescribed in the leucosantha clade, and we also detected gyromitrin in that species. We tested more Gyromitra splendida because from the paper, we talked about how we detected less gyromitrin in splendida than we did venenata, which actually fit with a hypothesis that people had. It was kind of this anecdotal hypothesis that West Coast Esculenta group species had less gyromitrin than the ones on the east. And we found that in one specimen, but we tested more after that. And we found that they actually had large amounts of gyromitrin. So they doesn't seem like they contain less than venenata. And then there's Gyromitra anthrocobia, which is the most closely related species to the Esculenta group. Anthrocobia is a very strange species. It has some really it's very similar to some Morchellaceae species, like Verpa species. It doesn't quite look like a Gyromitra so much. There's only two collections known from the world, one where it was described from Cyprus, and then another from the Pacific Northwest. Leah Benlin from the Pacific Northwest shared that specimen with me, and we were able to get a culture of that, and that one tested negative. I thought maybe it would test positive. So that's some other new data for our tree. Great, so let's move on to the discussion, and where we'll talk about, I'll summarize what we've learned from this research and discuss the edibility of Morchelles, and then share some of the ongoing research that we have in this group. So what we found is that gyromitrin has a limited and discontinuous distribution in the Discinaceae family. It was present in all tested specimens of the Esculenta group, and to our surprise, in all tested specimens of the Leucosantha group. Assuming that gyromitrin is indeed present in Gyromitra gigas from Europe, there would be, it would require many loss events, loss of this trait, to explain its distribution in the family. And it's unlikely, therefore, that it's a product of inheritance from a common ancestor. More likely, this distribution is a result of horizontal gene transfer, which is not uncommon with secondary metabolites. It's also not unheard of for there to be many losses of a secondary metabolite. So currently, we don't have enough information to understand exactly how it's evolved in the family. A caveat of all this research is that gyromitrin could be present at concentrations below our detection threshold, or there could be intraspecific variation in gyromitrin production. This was found in that paper I mentioned from 1980, where they found gyromitrin in very small quantities in 2 out of 3 specimens tested. For our biological and research purposes, we would like to know if specimens have very small amounts. Fortunately, for people who are eating these, if the quantities are below our detection threshold, that shouldn't have any impact for human health. And however, despite the caveat, the consistency of gyromitrin in a group that contains gyromitrin, as well as its longevity, is inspiring. We think that if it was present, we would have detected it. And now talking about the edibility of Morchell's, and I like to talk about this. This is why I got interested in this group in the first place. My advisor someone reached out to him talking about a proposal for working on these Morchell's. And I said, sign me up. If I can go out in the spring for my PhD and collect morels, that sounds great. I'll do that. That's how this project started. And this research is informing people's understanding of the safety in terms of eating Morchell's. So my thoughts are that no one should be eating these gyromitrin containing mushrooms in the Esculenta group or the Leucoxantha group. I think it's not worth the effort, as well as we don't have a good sense of the long-term health impacts. There are the methods to process these mushrooms to not be poisoned by them in an acute sense, but we don't know the chronic long-term exposure effects of exposure to gyromitrin, which could include neurodegenerative disease. So I would recommend to avoid any mushroom that contains gyromitrin. Then there are these Morchell's for which there is already a culture of consumption, and these seem okay. These include the North American Gyromitra gigas group, as well as the Brunea group. I say North American because there's this European Gyromitra gigas for which gyromitrin was detected in the 80s, and we need to follow up on that and see if that is still the case. These mushrooms, there's no evidence besides not containing gyromitrin from our tests, our specimens. There's no evidence of extraordinary toxicity. And I say this because you can find reports of people getting poisoned by them, but the poisonings are no more severe than mushrooms that people consume and call edible otherwise. Things like morels. Morels are known to cause poisonings. There's lots of ways in which someone can get sick from eating a mushroom. They can be old, they can be misidentified, or someone can have an allergic reaction. So with that caveat, there's actually no evidence for any of these mushrooms being more toxic than mushrooms that people typically eat and regard as edible. So if people are to eat these mushrooms, it's absolutely important that you positively identify the species you're eating. You collect good mushrooms, not ones that are rotting, and you cook those mushrooms thoroughly. Morels, for example, people eat all over the place. However, if you eat them raw, people do get sick. It actually can cause strange neurological symptoms like shaking and confusion. And we have no idea what toxin or what substance in those mushrooms is causing that symptom. But if people cook those mushrooms thoroughly, they're safe to eat. However, with any of these mushrooms, and this includes morels, this includes chicken of the woods, this includes pretty much any mushroom you're foraging out there, there are dozens, if not hundreds, of unidentified unique compounds produced by any group, like the Morchell's. And these are typically entirely uncharacterized. The world of fungal chemodiversity is vast, underexplored. These substances have great impact. Many important substances have come from fungi, but even just in these common species, there's just so few of them have been evaluated and characterized. It's an extremely large amount of work to do so. However, with the Morchell's, there has been one substance that has been characterized from Gyromitra fastigiata. This species is closely related to Gyromitra brunea. It's found in Europe. And researchers in the 1980s found this chemical called 1,2-hydroxyacetylpyrazole. What they found was that a similar molecule, it wouldn't take much for a similar molecule to turn into gyromitrin. A hydrolysis reaction of the pyrazole could result in gyromitrin. So this might give pause to people who are trying to eat Gyromitra brunea, the sister species. Even though we haven't detected gyromitrin, it's true for all these mushrooms that people are eating that it's just a completely unknown world of chemodiversity. And some ongoing research we have following up on this study, we are getting some European species, including Gyromitra gigas, and we are going to test those to see if that has gyromitrin, which would change pretty drastically how the evolution occurred in the family. And we've done a lot of whole genome sequencing. I have 50 genomes sequenced from the family. I've used phylogenomics to infer evolution of the family. Forever with molecular phylogenetics, we have been not sure of the basal relationships between the clades. And with our phylogenomics, we've been able to understand how the clades are related to each other. And this is going to result in a number of taxonomic revisions, not only or beyond clarifying the evolution to allow us to infer an accurate species tree in understanding the evolution of Gyromitrin. But the real holy grail of my research is identifying the actual genes that produce Gyromitrin. To do this, we're using comparative genomics and transcriptomics, and we'll be using gene knockout experiments to confirm their role in toxin production. Once we identify the genes, we'll be able to look through the genomes of the family and really get a sense of which species have the capacity to produce Gyromitrin or perhaps a related chemical. With that, I want to say thank you for your time, for listening to my talk. I want to remind you, please support Asian Americans United if you have the means, as we work to oppose the arena and protect Chinatown. And if you have any questions, I would love to take them now.
Thank you so much, Alden. That was great. Yeah, again, if you guys have questions, feel free to write them in the chat here, and we'll read them off. Also, if you use the raise your hand feature, we can call on you and you can read your question out yourself, or speak your question. So you already have one. That's awesome. Did you test for Gyromitrin presence after double boiling technique, or will you? And did you see any differences between Midwestern Esculenta and West Coast Esculenta?
Great. Yeah. So we didn't do any processing of the mushrooms beyond drying them to preserve them. There have been a lot of studies from Europe in the 1970s and 80s where they were doing those kinds of tests to see within Gyromitra esculenta how much Gyromitrin is lost. So yeah, I'm not replicating that kind of work to see the rate at which it's lost with techniques. Yeah, just focus right now on presence and absence, and I hope others will follow up with this research to actually figure out the toxicology. I certainly don't have the expertise, the medical expertise to evaluate how much Gyromitrin is too much Gyromitrin. So personally, for myself and for others, I recommend just don't mess with any of these mushrooms that contain the toxin. We're far from understanding its full impacts on human health. But I hope others will pick up this research to understand exactly what kinds of concentrations we're working with different specimens from different regions. So with the question of differences between Midwestern versus West Coast, at first we thought that the West Coast one specimen that we tested had less Gyromitrin, but we tested a number of other ones after that, and it doesn't seem to be a big difference. But this is with dried specimens. You get differences, idiosyncratic differences just based on how they're dried or whatever. So yeah, we're not in a position to really say the concentration that's made by different specimens from different biogeographic areas or regions.
We have a few more questions here. Is there any possibility that symbiotic microbes might be involved in toxin synthesis?
Yes, there is a possibility of that. Theoretically, I don't think realistically, because I have cultures of Gyromitra venenata, for example, and it produces Gyromitrin. We sequence the genome and there's no DNA of anything else. It's just fungal DNA. For that reason, I think it's unlikely that it's a symbiotic microbe. However, that is a thing that happens elsewhere. But yeah, that's the big question I'm trying to figure out right now, is just like what genes might produce this substance. And it's challenging. So any ideas is great, because I'm really struggling to figure out which genes to target in our knockout experiments.
Yeah, I was actually going to ask about this as well, because excuse me. So I've done a little bit of work with lichen chemistry as well, and looking at specific genes, things like that. And your study reminded me quite a lot of some things that we found in our research with cladonia chemistry, which was quite interesting. And we're kind of seeing that if the gene clusters that are responsible for the production of that compound are present, they're like always kind of working, even if they're making things at very low concentrations, like concentrations too low to be detected by some methods, but not mass spec. And then in other species, the genes are just missing. And so actually what you said towards the end there about how these metabolite genes in fungi kind of jump around because of horizontal gene transfer, that was very interesting to me, because that's sort of what we had been seeing in our lichens too. I was actually wondering if you knew anything about the mechanisms behind that.
Oh yeah, that's a great question. Horizontal gene transfer is a fascinating thing that it's increasingly being appreciated for its role in evolution. I don't know how it exactly happens. My imagination says that cells are leaky, and over millions of years, with all the trillions of individuals on this planet at any point, all the microbes and everything interacting, like DNA gets around, it gets up into cells somehow, and chance events lead it into being incorporated into the genome or being expressed. And when you have something that's really valuable, it sticks around. I don't know if that's how it works, but that's how I imagine it to work. And I think biology is so vast and time is so vast that even if it was like a spontaneous and chaotic thing like that, then it happens enough times. With these secondary metabolites, they are, the consensus is that you do have really rapid evolution along with loss events and horizontal transfer. And that's because these secondary metabolites are very valuable, but they're also costly to produce. So, and especially if you're in a community where others are producing metabolites that are useful for you, you don't need to produce that thing. Or if you want to focus on other aspects of your metabolism, you might no longer produce a secondary metabolite because it's just not doing enough for it to stick around in the process of evolution. So you get these, it's really common, even within a species, you can have individuals that have the genes to make a secondary metabolite and ones that have lost it. So it's a quite, it can be all over the place. And I think we're only at the very beginning. Hopefully with our genomes, we can start to figure out exactly how it's spread across the family.
That's very interesting. I have one more comment on that before I turn it over to the next question. It's just that the idea that if there's redundancy in specific chemicals that are being produced and secreted into the environment, if someone else is doing that, you don't have to do that. You're sort of free to evolve away from that. I find that very interesting. I believe that's this Black Queen hypothesis idea in evolution, where you could potentially trap another species into a costly ecosystem service. If you both have the capacity to do that, but then you lose the ability first, you sort of trap the other one in that important responsibility that benefits both of you.
Yeah, exactly. Yeah, that's an important thing. And in addition, something I should mention is that, sometimes, oftentimes secondary metabolites are used in ecological interactions, and that's kind of primarily what they're thought of. They're extremely important in interactions with other organisms in the environment. However, some of these things are not necessarily secondary metabolites in that sense. It could be that some of them are just breakdown products. And right now I'm exploring the idea of whether Gyromitrin is just a breakdown product. If you have toxins that develop in your self, including these non-proteogenic amino acids, you might have enzymes that break those down. So there's a lot of reasons why these small metabolites might be being made, and they could be being made for extremely very specific ecological reasons, and sometimes they're just made because it's a breakdown product.
Awesome, thank you. Another question for you: is the genus mycorrhizal? And if so, is that symbiosis relationship linked to the production of Gyromitrin?
Yeah, that's a great question. I'm going to go back to this tree, which, I have knowledge that you do not, which is I have a phylogenomic tree that has clarified all the relationships of these species. Anyway, so I'll just say that this tree kind of shows that, but you have this bottom section here, the Venenata group, Ambigua group, the truffles. So this whole group, it's really interesting because I've gotten pretty much every single thing in this group to grow from spore. We have cultures of pretty much all these: Anthrocrobia, Splendida, Venenata, Infila, Ambigua. All of these we have cultures for, including a Hypnotria, which is probably mycorrhizal. That's on a special mycorrhizal medium. However, everything in this top clade, nothing has grown from spores. I haven't gotten anything to grow on plates from spores. So I think that there is some sort of ecological signal in the phylogeny of the family. I don't think it's mycorrhizal in the sense that these like venenatas they grow extremely well in culture. They have no problem with that. You find infila growing on wood directly sometimes, so that could imply a saprotrophic or decay ecology. But largely, we don't really understand the ecology of these species. But that's my thought right now, that there is something symbiotic happening with this top clade. Maybe that's an endophytic relationship or a mycorrhizal, perhaps. And that's why these things do not germinate in culture. Whereas this bottom clade, my thought is that these are saprotrophic, or perhaps they're in some sort of like weakly pathogenic or obligately or facultatively symbiotic relationship, such that they grow fine in culture. So that's a long answer to the short answer is that we don't really know the ecology of these species, actually.
Interesting. While we wait for more questions, I have a follow-up to that, in the sense about your culturing. I was curious, so you were able to get some of these species to grow from spore in culture. Were you also able to get them to fruit in culture?
No, I haven't. I haven't. That would, yeah, I would like that eventually, but right, that's kind of like the obvious next step to all this research, which is also why I've been excited about this. If we could actually cultivate these mushrooms and understand which ones are safe to eat or not, then we have a new mushroom crop, which would be really exciting and definitely a dream of mine. No, we haven't got any mushroom production. I haven't necessarily tried so much, but yeah, nothing, like definitely nothing from the plate. Sometimes you get that. There's a lot of other things I'd like to do before I can like understand exactly what they need. These ascomycetes have mating type loci, so we could look into their genomes to see if they need to have sex with another strain or if they're self-compatible, whether they need to sorry, I'm losing my words. If they need to, reproduce with another strain to make a viable dikaryotic mycelium to make a fruiting body, or whether they're self-compatible. And that will help us understand what they would need to actually get to the point of producing a mushroom. But yeah, I think we're pretty far from that point of actually cultivating them. In Europe in the 70s and whatnot, people were really motivated by this. They're motivated to understand which species had gyromitrin. They're motivated to try to cultivate them. And that research, I think people were limited by the tools available at that time. And then there was a long dry period since basically the 80s until now where no one worked on these mushrooms. I think that happens a lot in biology, where you have something that's in vogue and then it's out of vogue. So I'm trying to pick this up again because it's a really interesting system. People care about these mushrooms. It has impacts on public health, and it informs what we want to be eating and what we want to be putting into our bodies, and perhaps in the future, whether we can cultivate these or not, beyond the larger questions of genetics and evolution, which will have a lot of implications for finding other N2NPs and other genomes and whatnot. So yeah, it's an exciting group to work with after a long hiatus that it was on.
Yeah, for sure. And like the sort of, I guess, another dimension of this that I would find quite interesting is like where, if there is variation in where the compounds are being produced, because at least in some lichens, we often have situations where, like in the reproductive structures for said lichens, we have some chemical being produced that is a deterrent from consumption, or only in the vegetative material and things like that. There is some variation there. And of course, sometimes there's also like an environmental influence too. Like, there's been grazing that's been experienced by the fungus. And so it's triggered to produce these things. If the if you find the genes, which would be amazing, if you find the genes responsible for that, I would be very interested to see which of these groups, which of these different species have those genes, and do you find any ones that you didn't expect to have the genes that do have the genes? And then, like, you know, when are those genes active, or are they functional, and things like that?
Yeah, totally. You know, that would be a good first hypothesis around the functioning of this secondary metabolite. I said it could be a byproduct, but you know, I think the most reasonable hypothesis is that it's producing this toxin to reduce predation, to reduce mycophagy. Humans certainly are poisoned by gyromitrin, and just on the way it works, it's like the chemical basis of it, I would expect it to be toxic to any animal that was eating these mushrooms. So yeah, I think that's a reasonable first hypothesis for its function. And it would be really interesting to do some transcriptomics comparisons once we know what we're looking for between the mycelium versus the fruiting bodies. We know the mycelium produces it, but yeah, seeing if those genes are upregulated in the fruiting bodies, and or if they're upregulated after some sort of mycophagy event, would be a great way to test that hypothesis. So hopefully we can get there at some point.
Awesome. Well, I'm rooting for you, because this was super cool.
Thank you.
I don't see any other questions, so I think that we can close things out here. I thought this was fantastic. I hope that you guys all enjoyed this. Thank you, Alden, for coming to speak to Torrey Botanical Society. Really appreciate it. This was great.
Thank you so much for having me, and have a great night, everyone.